So last time we talked about clinical specimens and methods to collect them. So we talked about the different locations and how we collect them. Today we're going to talk about obtaining a pure culture in different types of media. So we obtain a pure culture, and a pure culture is composed of cells arising from a single progenitor. So this is one cell that forms a bunch of other cells. And the progenitor is termed as a colony forming unit, or CFU. And we can count CFUs on a Petri plate by how many different circles we have. So aseptic technique prevents the contamination of sterile substances or objects. So we perform aseptic technique to keep that pure culture pure. There are two common isolation techniques. We have the streak plate and the pour plate. So the streak plate, you start with your sterile inoculating loop, and you pick up some of your bacteria and you streak it in quadrant 1. It's also called quadrant streaking. Then you turn it, you clean your inoculating loop, and you swipe from 1 into a new 2. And you're pulling some of those colony forming units over to that new section. And then you repeat for 3 and 4 until you have isolated single colony forming units where you can count them. So you can see in the plate example, you can literally count how many colony forming units are there. You can also do the pour plate method of isolation. So you take your initial sample and then you subsequently dilute it until you get fewer colonies in that final pour plate. So you can see there's quite a few colonies before you dilute it that final time. And then you can count the colony forming units and you can see them differently. So we can see here there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 1, 32, 33. There are 33 different colony forming units in photo B. So we can also isolate some fungi with streak and pour plate methods. Protozoans and multi-unicellular algae are also isolated through dilution of broth cultures. So instead of pouring it into a plate, we just continue to dilute that broth. And then some of the larger microorganisms, we can pick out a single cell to establish a pure culture. Now that we've talked about how we set up a pure culture, let's talk about what we're going to do with that pure culture and the media that we're going to use to grow it. So culture media is super important because that's what we grow bacteria and other protozoans or prokaryotes or single cell organisms on in lab. So a majority of prokaryotes have not been grown in a culture media. That's because they have super specific requirements that we cannot replicate. So nutrient broth is the most common media that we will use. And then we have an additive called agar that we add to many of our media types to make it a solid surface. So agar is a complex polysaccharide that can be derived from a red algae, and it produces that solid surface of for colony growth. Most microbes cannot digest agar, so you do have to add a nutrient broth or another type of media in for that microbe to digest. So there are 6 types of general culture media. We have defined, complex, selective, differential, anaerobic, and transport media. So before we talk about those 6, I want to talk about what our solid media can look like. So we have Petri plates, which you've seen a picture of. We do have these things called slants. So slants have a slant at the top and a butt at the bottom. And the butt is the bottom part, and we can see different things happen to the butt and the slant depending on what kind of media it is and what we're using it for, which we'll talk about later on in this chapter. So the first type of culture media we're going to talk about is defined media, media in which the exact chemical composition is known. So we know exactly what goes into that media, and this is used for fastidious organisms or organisms that are super, super picky. That require an additional amount or an addition of a large number of growth factors. This is pretty selective, so one thing's gonna grow on it and the other thing's not. So defined media is very, very specific to the type of bacteria you want to grow. So here's a great example of that defined media. So we can see this defined synthetic media. We can pull glucose at the exact value. We can pull sodium potassium at the exact gram value. We can pull, and I can go make this with the stuff we have in our closet. And then we use distilled or deionized water to bring it up to 1 liter. So it is the exact amount that we need to make this exact media to grow E. coli effectively. So we have defined media. Complex media is where we don't know the exact composition of that media. Certain nutrients we use that are released by partial digestion of yeast, beef, soy, or other proteins. So this is going to be what we use most commonly for just keeping our bacteria alive in lab. This is going to be our tropic soy agar, or our TSA, or our tropic soy broth, or our TSB. It supports growth of a wide variety of microorganisms, and it's not specific to one. This is used to culture organisms with unknown nutritional needs. So if we don't know exactly what they need, we'll put it in a complex media like TSA so it can grow and not die. So you can see here is an example of a selective media. So a selective media is a media that is used to select different types of bacteria or fungus or algae based on their growth requirements. So if you know the bacteria you want to grow, like the pH of 7.3, then you're going to use an agar that is a pH of 7.3, so that bacteria can grow while nothing else can. So if you know that your colony wants to grow at pH of 5.6, so they want a more acidic environment, you're going to make an agar of a 5.6 so that bacteria will grow, and then you can, things that can't grow in that high acidity won't grow there. So you are selecting different things that you know about that make that thing grow or not, if that makes sense. You are selecting the requirement that you want and pushing everything else aside. Enrichment media is where we take a selective media and we use it to increase the number of chosen microbes to observable levels. So once we get a microbe we want to look at, we can then take a selective media and start increasing the number of that microbe to where we can look at it more. So this may require a different series, like a series of cultures to enrich the bacteria to a desired amount. One example of this is cold environment. So if we know the bacteria we want to enrich is cold tolerant, we can take that bacteria and put it in a cold environment and help it to enrich the area, while things that aren't cold tolerant won't be able to survive there. So here's another example of differential or selective / differential media. So this is called blood agar. And what blood agar does is differentiate between bacteria that will ingest and digest blood and bacteria that doesn't. So if they are beta hemolysis, that means they digest blood. So you're going to see this white coating around it, almost yellowish or clear. Alpha hemolysis, they kind of can digest blood, but not really. You're going to see this greenish color. And then if no hemolysis will happen, or gamma hemolysis, so they cannot digest blood at all, you're going to see the solid form. We can also look at a differential media here. So we put a bacteria within a carbohydrate fermentation test, and we can see if they will either fermentate carbohydrate or not. So the first bacteria obviously did not perform any fermentation, while the second bacteria did. So you can see the media changed color, and we were able to trap that gas bubble to see that bacteria produced gas. We can do it also here with monkey koi agar. So monkey koi agar is a selective and a differential media. So we can have a nutrient. Our first example is a nutrient agar. So Staphylococcus aureus and E. coli will grow on nutrient agar. But when we put Staphylococcus and E. coli on monkey koi agar, Staphylococcus aureus isn't going to grow, but E. coli will. So if we then take E. coli and Salmonella, we can see they both produce different colors, meaning one ferments lactose and the other one doesn't. So E. coli ferments lactose while Salmonella doesn't. So it is telling us that one does something, the other one doesn't. So it's differentiating between it. And then we can also see one grows on it and one doesn't. So it's selecting our different bacteria. So like I said here's our representation of differential media. So monkey koi media and blood agar. The thing I want you to remember from this lab is monkey koi agar is for differentiation and selectivity for bacteria based on the ability to ferment lactose, while blood agar differentiates between bacteria that can digest blood. So we've talked about differential media, selective media, complex media. We are now going to move on to anaerobic media. So anaerobic media is media that we use to grow obligate anaerobes. And they must be cultured in the absence of free oxygen radicals, right? Because free oxygen radicals are going to damage the cells and not let them grow. So reducing media containing components that are combined with free oxygen and remove it from the media. So we are going to take anything that contains those free oxygens, pull it out of the media. Petri plates are incubated in an anaerobic chamber vessel. This is a sealable container that contains reducing chemicals. So you can see it here. We have an anaerobic culturing system. It has a clamp, has a chamber. The lid is airtight. We have paddle pellets to catalyze reactions removing O2. We have an envelope that contains chemicals to release CO2 and hydrogen, our Petri plates, and methyl blue, which is our anaerobic indicator. Then we have transport media, and this is used by hospital personnel to ensure clinical samples are not contaminated and to protect people from infection. Wow, English. Rapid transport of samples like this is super important because we don't want the sample to degrade or lose its function. And then we also don't want the samples to get out and infect people. So there are different kinds of culturing techniques. So techniques are developed for culturing microorganisms, but we can also culture animal and cell cultures. So we can use animal and cell cultures to grow bacteria where artificial media is inadequate. So we typically use animal or cell cultures for growth of viruses and other obligate intercellular parasites that we can study. So these are things that we cannot grow in culturing media that specifically need animal cells or an animal to grow in. So we can look at them. We also have low oxygen cultures. So many organisms prefer intermediate oxygen levels. So they still like oxygen, but not as much as out in the regular world. So we can use carbon dioxide incubators to mimic the environment of many different body tissues. So we actually have a carbon dioxide incubator. It's our big incubator in the back. I just have to hook it up to a carbon dioxide tank. But there is a low-cost alternative called a candle jar. And these are ideal for growth of canophiles or microbes that grow best in high carbon dioxide levels. So you put a candle in a jar, you light the candle, oxygen leaves, carbon dioxide stays. So we will end here. We will finish with preserving cultures tomorrow. We will take a test on Friday, and then we will start lab Tuesday next week.